This video is about an environmental engineering and microbiology project involving the movement and quantification of pathogens in a managed aquifer recharge site. My name is Hannah Collins, and I am a student in the Summer Research Internship program at Arizona State University. I am majoring in environmental engineering. The goals of environmental engineering are to protect and enhance public health and environmental quality, which are also the goals for this project. The objectives for this project are to quantify pathogens in the environment and to characterize the movement of pathogens through the environment. The pathogen of focus is E. coli, and the managed aquifer recharge site of focus is the water reclamation facility at the Riparian Preserve at Water Ranch in Gilbert, Arizona. This project is a combination of two current projects at Arizona State University, one from Dr. Rebecca Munich's lab and one from Dr. Carrie Hamilton's lab. The project in Dr. Munich's lab is quantifying trade-offs in water quantity and quality in managed aquifer recharge systems. Water recharge sites are often used to replenish reclaimed water that is pumped from the ground and to store surplus water in recharge basins or ponds. There are 7 of these ponds at the Gilbert Riparian Preserve. One of these ponds, Pond 7, is shown in Image 3. There are both benefits and risks to using water recharge sites like the Gilbert Riparian Preserve. One benefit is that these sites reduce demands on groundwater use. Providing an additional water source for a rapidly growing community like Gilbert is important, especially in an arid environment like Arizona's. Another benefit is the creation of desert riparian habitat that attracts a variety of wildlife and promotes biodiversity. The riparian areas, including the site in Gilbert, only take up less than 1% of the land in Arizona, and yet this land supports more than 60% of the wildlife in the state. The risks involved with the use of MAR sites have to do with chemicals and toxins, as well as pathogens such as E. coli, which are commonly found in wastewater due to fecal pollution in the water. Dr. Hamilton's project focuses on antibiotic resistance in the environment. The risk to human health is significant when it comes to water and wastewater environments, since antibiotic-resistant bacteria and genes thrive in them. When medications are no longer able to kill bacteria, it is a very significant problem, especially since drug-resistant bacteria can transfer a copy of their genes to other bacteria that need those genes to survive. This process is called horizontal gene transfer. The adaptation, evolution, and survivability of pathogens like E. coli is further helped in biofilms, where the bacteria stick together, exchange DNA and nutrients, and undergo changes to evade the immune systems of humans and animals. Antibiotic resistance and horizontal gene transfer in the environment make pathogens like E. coli difficult to manage. The two projects come together through the connection between E. coli and its health risk through effluent sources. E. coli bacteria are commonly found in the intestinal tracts of people and animals. They are also found in foods and in the environment. E. coli's presence in water samples is an indication of fecal pollution. This can make people sick with a variety of symptoms which differ in severity. Understanding how E. coli is transported in the environment can aid the community of Gilbert in avoiding harmful exposures to the pathogen, which can happen through direct skin contact, inhalation, and/or accidental digestion of the wastewater. For the two projects in Dr. Munich's lab and in Dr. Hamilton's lab, understanding both how horizontal gene transfer impacts antibiotic resistance and how porous media, such as sand or soil, impacts the fate and transport of pathogens is key to protecting public health from exposures to pathogens by preventing them from happening. The Gilbert Riparian Preserve was constructed in 1999 to replenish groundwater supplies and support native riverside forest and wetlands where wildlife can live. These wetlands were created by the water recharge basins, or ponds, that are shown in Images 7 through 9, as well as in a map of the Gilbert Riparian Preserve in Image 10. An effluent flow of wastewater feeds into each of these 7 ponds on a rotating basis, depending on when each of the ponds need to be dried so that tilling can be done to unclog the pores in the soil. Unclogging the pores is important for preventing the buildup of contaminants and nutrients in the soil. Too many nutrients can lead to algae growth, as shown in image 9. Algae growth can eventually lead to algal blooms, which have toxic and harmful effects on people and animals. Other than the 7 ponds, there is another water source labeled Water Ranch Lake in the map. The water that runs into the lake is groundwater, not wastewater like in the ponds. People are allowed to fish out of Water Ranch Lake, but not in the 7 ponds due to pathogenic intake risks. Despite that the wastewater from the effluent is treated, it is still not potable or drinkable. So how do managed aquifer recharge systems and E. coli come together in this project? The project's application to the Gilbert Riparian Preserve lies in how water moves through soil. When the treated effluent wastewater enters a pond in the Gilbert Riparian Preserve, the water and the E. coli in the water seep deep into the ground and can spread all the way to another water source. This is a problem when the E. coli from one of the ponds, like Pond 7 for example, infiltrates into Water Ranch Lake, which is used recreationally. To see how much E. coli has infiltrated into Water Ranch Lake from the ponds, a procedure called MTEC is used. MTEC stands for Membrane Thermotolerant E. coli. It is an experiment that is done to determine the concentration of E. coli in water samples. First, the day before sample collection, agar plates are prepared by mixing the appropriate amount of agar with water. The media is then autoclaved to sterilize and decontaminate it using high-pressure steam. Then 15 milliliters of the agar media is pipetted into each of the plates needed to process the water samples. Once the media solidifies, the plates are inverted and stored in a refrigerator. The next day, as soon as the water samples are taken at the Gilbert Riparian Preserve, they are stored in ice coolers and immediately taken to the lab so that the microbes do not die out before they are processed. In the lab, each of the water samples are filtered on a white gridded membrane filter. After the water has completely run through the funnel, the manifold, and the flask, as shown in image 10, the filters are each rolled onto the agar in a petri dish, which is then incubated, first for 2 hours at 35 degrees Celsius, then for 22 hours at 44.5 degrees Celsius. After taking the plates out of the incubator, you can see that on some of the filters of the plates, including the ones shown in image 13, there are little dots that appear to be magenta in color. Each magenta dot on the plate is an E. coli colony. Any colonies that are not magenta, like this white one, are different types of colonies than E. coli and are not included in the final results, which are reported as E. coli colony-forming units per 100 milliliters of sample. In the results, the goal is to have between 30 and 300 E. coli colonies on one plate. If there are more than 300 colonies, then the sample is labeled as TNTC, which stands for too numerous to count. An example of TNTC is shown in image 14, which is a sample taken from Pond 7. As you can see in the results, the Water Ranch Lake groundwater samples have significantly less E. coli concentrations than Pond 7's wastewater samples, both for the 1:10 dilution and the 1:100 dilution. However, the presence of E. coli in Water Ranch Lake is a cause for concern, especially when there are upwards of 67 colonies found in a diluted sample of only 100 milliliters. These concentrations indicate that there is subsurface spread of E. coli from the ponds to the lake. The second experiment for this project is with soil columns. The purpose of a soil column experiment is to characterize the movement of water and therefore E. coli through the soil. For these experiments, soil is packed into a column and water is pumped through the soil in the column. Both the soil and the water samples are collected from the Gilbert Riparian Preserve and are prepared and processed in the lab. The soil is collected in a dry area in Pond 7 using a macrocore soil sampler. After taking the soil to the lab, it is dried in an oven for 72 hours at 160 degrees Celsius. Once dried, it is broken up into smaller particles using a mortar and pestle, as shown in image 17. These smaller particles are filtered through a sieve, resulting in what you see in the bottle in the right side in image 18. The soil is then pasteurized by being moistened and spread out in a clear polyethylene bag 2 to 3 inches thick. After leaving the bag in the sun for 4 to 5 days, any weeds or insects that could have been in the soil were killed by the heat. After drying the soil in the oven again for 72 hours at 160 degrees Celsius, the soil is completely prepared to be packed into the column. The water that runs through the column is spiked with E. coli to match the conditions in the Gilbert Riparian Preserve as closely as possible. A soil column is helpful for researching water and pathogen movement in the environment, since it is a controlled system correlated to water content, soil particle size, and the column's diameter and length. For the column we are using, there is a 1:5 ratio for inner diameter and length. This is because if the column is much longer than it is wide, like for a 1:6 ratio or greater, then the gap between the porous media and the inner surface of the column could create a preferential flow path rather than being closer to the conditions you would see in the natural environment. Having a shorter column is also preferred since the E. coli can run quicker through the column. Using a column and setting it up vertically rather than horizontally helps to control the flow rate of the water passing through. Below is the experimental setup for the soil column experiments in image 20. The reason that the outlet is on the top of the column and that the inlet is on the bottom is because, due to gravity, it would be more difficult to control the flow rate if the water was passing from top to bottom. The water in the container to the right side of the setup is spiked with E. coli cultured in the lab and is pumped through the column using a pump with standard pump heads. The pump pushes the water through the tubing and column at a rate of 5 milliliters per minute. At the top and bottom of the column are stoppers that connect the tubing and the ends of the column together. The stoppers are sealed using electrical tape to prevent water from leaking. Attached to each of the stoppers inside the column is a filter to prevent the soil from running through the tubing while allowing E. coli to pass through. As the water exits the column, water samples are collected in tubes using a fraction collector, which rotates to the next tube every 3 minutes. The water being pumped into the column holds 10 grams per liter of potassium bromide, which is used as a non-reactive tracer to plot the movement of water and therefore E. coli through the column. This involves testing the salinity and conductivity of the water in each of the test tubes from the fraction collector using the probe shown in image 21. The salinity and conductivity are therefore categorized by time intervals as the water samples flow through the column. When plotting the results on a graph, the goal is to have a breakthrough curve that is shaped like an S, like in the graph on the left. The flow rate is an important component of the experiment since the soil conditions the flow of the water through the column, and the breakthrough curve depends on the flow rate. If there was no soil in the column to condition the flow rate and only water, then the graph would look more like a linear function. If the soil column's breakthrough curve looks more like the graph on the right, then that likely means that there is too much pressure on the column, since the pressure forces the water into easier pathways and creates preferential flow paths inside the column. This S-curve graph was generated from an experiment done with sand rather than soil to become familiar with the column experiment process before experimenting with the soil from the Gilbert Riparian Preserve. Each of the points on the graph are the conductivity and salinity measurements for each test tube. Conductivity and salinity are closely related because salts like potassium bromide, when dissolved in water, release a positively charged cation and a negatively charged anion. These ions conduct electricity. This experiment is a good start, but there is still evidence that there is preferential flow in the column since, based on our calculations for pore volume, it should have taken 75 minutes for the water to travel through the column, rather than only 15. The experiment overall was still successful in terms of gaining experience with column experiment test runs. In the future, we plan to obtain more data for the potassium bromide tracer once we run column experiments with the soil from the Gilbert Riparian Preserve. Other future work that can be done for this project includes quantifying antibiotic resistance of E. coli cultures, quantifying various antibiotic resistance gene concentrations, and integrating the quantifications into a risk analysis model to further the research and prevent the spread of antibiotic resistance in the environment. This project has strong ties with environmental engineering, which is what I am currently studying at Arizona State University. The main goals of environmental engineering are to protect and enhance public health and environmental quality, both of which are also the goals of this project. Looking at and investigating the movement and behavior of E. coli in the water and soil samples from the Gilbert Riparian Preserve is essential for predicting the fate of this pathogen in natural subsurface environments, assessing contamination potential, and developing efficient water treatment techniques to prevent exposures. A big thank you to my lab partner, my mentors, and each and every one of my colleagues who have greatly helped me with this project. And thank you for watching.